My name is Alia Aziz from Institute of Medical Science Technology, University of Kuala Lumpur. Today I would like to present my research project on development of in vitro neuronal injury model via overexcitation of kinetic receptor. Glutamate toxicity causes motor neuron degeneration, including spinal cord injury. Previous study has been mainly focusing on MMDA and also AMPA receptor, and little is known about kinetic receptor excitotoxicity. Overexcitation of glutamate receptor is an important pathway to neuronal cell death. Therefore, identification of cell death pathway provides an important overview to the pathophysiology of neuronal injury. The aim of this project is to develop an in vitro spinal cord injury model via overexcitation of kinetic receptor, with the objectives are to differentiate NSC34 cells into neurons via retinoic acid, to induce injury to cultured cells using high concentration of kinetic acid, and lastly is to identify the mechanism of cell death via quantitative real-time PCR. So basically, the NSC34 cell were cultured on poly-LIC coated plate and incubated for overnight. The next day, 1 micromole of retinoic acid was added to the culture to differentiate the NSC34. Then the injury were done by exposing the cells to kinetic acid, and validation of the neural injury were done by using observation by phase control microscopy and also cell death assay, and lastly by real-time qPCR. Moving on to the result and discussion, the first one is to differentiate NSC34 cell using retinoic acid. There are two conditions. A is NSC34 cells cultured in serum debrief media without retinoic acid for 7 days. B, condition of NSC34 cells cultured in differentiation media containing 1 micromole of retinoic acid for 7 days. As you can see in the differentiated media, there are more neural outgrowths can be seen as represented by the red arrow. Second result is on the injury via overexcitation of kinetic receptor. We have the result for phase contrast image. So the condition in A is the differentiated cells without kinetic acid, which is the control. B, differentiated cells induced with 0.5 millimole of kinetic acid for 48 hours. And C is the differentiated cells induced with 1 millimole of kinetic acid. So as the concentration of the kinetic acid increase, more clumping of dead cells can be seen, as shown by the red arrow. Cell viability were measured by measuring the number of healthy cells in different concentration of kinetic acid. The third result is on the mechanism of cell death. We observe the expression of RIPK3 and also caspase-3 using quantitative real-time PCR. The bar chart shows the expression of RIPK3 and caspase-3 following treatment with kinetic acid at 0.1, 0.5, and also 1 millimole of kinetic acid. In conclusion, NSC34 cell cultured in differentiation media containing 1 micromole of retinoic acid show more neural outgrowth after 7 days in comparison to the control. When 1 millimole of kinetic acid added, cell clumping can be seen and neurites were disintegrated. This indicates cell death. On the other hand, the differentiated NSC34 in control condition retain typical motor neuron-like structure with intact neurites, which indicate the healthy cells were unaffected by kinetic-induced injury. And also the result from cell death assay shows that approximately 60% of the cells were viable in the condition of 0.5 and also 1 millimole of kinetic acid. And also to study the mechanism of cell death, gene expression of RIPK3, which is the marker of necrosis by programmed cell death, and also caspase-3, the marker for apoptosis cell death, were quantified. And it shows that the expression of RIPK3 was significantly altered by approximately 7-fold as compared to the control. Whereas for the caspase-3, there is no significant changes of expression in all concentration. So the result suggests that neurons die due to the overactivation of kinetic receptors via necrosis by programmed cell death. This finding can be useful in the future to further extend our understanding on the molecular mechanism underlying spinal cord disorder, thereby identifying new therapeutic targets. Here is